Thank you very much. So thank you to all of you for the invitation. Philippe, colleagues, Ivana, who was very efficient with all this preparation, and to IHU Mediterranean Infection Group. Thank you very much. It's a pleasure to be here. I know some of your work on this, and I will summarize for you today Quasispecies, basically an introduction to the concept, implications for the control of viral disease, and I will also mention some of our recent works. So let me begin. I said at the beginning that the sentence at the top is just a kind of, I want to transmit the idea that virus evolution now is very important to understand the emergence of viruses, disease mechanisms, and planning of virus control strategies, which a few decades ago, that was not obvious. Virus evolution was something separate, let's say, from the rest of problems that interested virology. So my talk today will be definition of quasispecies, mutation rates, mutant spectra, and consensus sequences, then fitness and the effect of bottlenecks, parallels between cancer and virus dynamics, quasispecies and antiviral strategies with some of our recent work with SARS-CoV-2, and finally, a slide on conclusions. So you are very well aware that what we mean by quasispecies are these complex mutant distributions that occur unavoidably inside any infected host with animal or plant RNA viruses. So in this slide, what I depict here is an infected virus with a line, and then what we call a mutant spectrum, which is the progeny of the virus that infected the organism, in which individual mutations are shown by color symbols. Okay, so mutant spectrum, these lines, you have to imagine now this is much more complex because each infected individual may have 10 to the 10, 10 to the 11, 10 to the 12, in different compartments and so forth and so on. So the situation is extremely complex, but that's the way we depict quasispecies as these mutants, these mutant distributions that are always subjected to change, competition, selection, and so forth and so on. And one of the basic molecular mechanisms of such diversity are the high mutation rates of RNA viruses, 10 to the minus 4 mutations per nucleotide copied. Okay? So this is the key fact, and that's what determines that mutant distributions are the norm, not the exception during an infection. Now, in addition to mutation, as you well know also, there is recombination in RNA viruses. Of course, DNA viruses as well, but in RNA viruses, recombination varies depending on the virus group. And then we have reassortment. The typical example is influenza virus, and I have drawn here an example with the 8 segments of influenza virus. And I want to say that this mutation, recombination, and reassortment are compatible mechanisms of genome variation. So you find these things in most RNA viruses that one has studied today. An example of how a mutant distribution looks like, and I'll show more of this later, is, for example, in one patient, we are studying several patients now from essentially from Madrid, from Spain, individual isolates of SARS coronavirus 2. And the typical image that we see, this is a shot, a part of the spike coding region. But what we see, this is the typical, that you see each line is a different sequence with a different frequency that is difficult to read, but you have a sequence with different frequency. But you see scattered a number of point mutations and also deletions. And we do that now by ultra-deep sequencing. And with this, now we usually have a range of reads per amplicon per unit of amplified segment, that the number of reads goes between 30,000 and 250,000. and we reach a resolution of about close to 0.1% frequency cutoff. And I'll justify this because that we have had some difficulty convincing some people that this level of resolution was really accurate, but I'll show some data to prove it. So that's the typical thing that you see by deep sequencing. When you see an isolate, you amplify, you go to the machine, and then you see these scattered mutations and so forth. Now, let me make some kind of conceptual introduction. This slide tries to show you how we can look at variation and what we can do experimentally to understand virus evolution in a better way. So what we can do to quantify variation is we take a sample from an infected individual, and if it's an RNA virus, we do RT-PCR. And if we put this directly to the sequencing machine, we obtain a consensus sequence. But if we have a way to look at the individual genomes of the samples, then we can get the mutant spectrum composition, as I showed diametrically in the first slide. So we can do that classically with molecular cloning and Sanger sequencing, and we have a limited number of sequences per genomic region. But nowadays, fortunately, we have deep sequencing, and we can get 30,000 to 250,000 sequences per genomic region. So that's the way we can learn, and we have learned about diversity. And of course, with the virus, once the virus can be isolated, if the virus can grow in cell culture, I don't want me to extend on this, but you know very well that we have accumulated a lot of data over the last 20, 30 years about biological clones, passages, persistent infections, serial passages in the presence of drugs, antibodies, etc. And we can see the evolution of the virus in the laboratory. So these two things, the increased potential to analyze individual isolates, natural isolates, and what we can do in experimental evolution have kind of contributed to what we know about virus evolution at the moment. So I like to distinguish the implications of quasispecies in three groups. One on the left in blue, the most theoretical, and on the other side, the green, the most, let's say, medically relevant application. So among the most theoretical is that the work by Manfred Eigen and Peter Schuster on the theory of quasispecies has told us that the wild-type virus, as was indicated in the old textbooks of microbiology, virology, the so-called wild-type virus is not a sequence. It is a set of genomes. Okay, so we have to start forgetting about a virus as a genome with a sequence, no matter what the textbooks say. And that blue part of the theoretical implication of wild-type has several interesting implications, and one is that the genome ensemble is the one that may dictate behavior, not always, but sometimes it may dictate behavior, and that for several purposes, the consensus sequence is a simplification and not sufficient to interpret at least some types of host-virus interactions. Now, in the middle, you see the key finding in terms of adaptability, and that is that quasispecies gives you a potential of what we call exploration of sequence space. In other words, viruses, due to quasispecies, can be at different places at the same time. I'll mention that now in the next slide. And that gives the potential for strong evolutionary transitions. And from the point of view of control of viral disease, essentially, we have learned over the last even 20, 30 years that now it's not possible to think of a vaccine that is just a synthetic peptide, or it is very risky to give monotherapy for a viral infection, because we have to confront the virus with different selective pressures, and that requires combination therapies. Okay? And final implication in terms of control of viral disease is what we have been working quite a bit in the last 10 years, 10, 20 years almost in my laboratory, which is virus extinction by crossing an error threshold of what we call lethal mutagenesis. So as a summary, evolution can be viewed as the replacement of viral subpopulation by others, and a consensus sequence hides phenotypic potential of viral populations. And that's shown in the next slide. And what I want to mean is that even if the mutant distribution that you have on the left has a certain behavior, inside it may be suppressed, but inside there are genomes that have the potential to behave differently. There are several here. They have been reviewed. I don't want to go into details, but I have underlined 4 of them. Partial resistance to inhibitors, antibodies, or CTLs. That's the justification of combination therapies, as I mentioned a minute ago. And these resistant escape phenotypes, you may not see them in the whole population, but they may be a minority in the mutant spectrum. Also, we discovered a number of years ago that there is a molecular memory in Quasispecies. And that means that if a virus has gone through a selection previously, even if that genome that is selected, then it can be diluted by replication of the Quasispecies, it remains at a higher level in the Quasispecies. We call this molecular memory, and the implication is that if you give a treatment, for example, to a patient, and the viral load goes down, but then you remove that treatment, you may have resistant mutations, resistant genomes remaining as part of memory. Another thing that has been documented in some cases, that is not so easy to document, but in some cases, there has been a switch between acute and persistent infections due to fluctuations in the Quasispecies. And obviously, you can have viruses that are virulent in an ensemble that is non-virulent, or vice versa. And that was shown classically very nicely by the work of Konstantin Chumakov with the poliovirus vaccine, that even if the poliovirus was attenuated, inside the attenuated vaccine, there were virulent variants. And these virulent variants, if they went above a certain proportion in the Quasispecies, manifested the virulence. So genome subsets may differ from the ensemble in critical phenotypic traits. So mutant spectra are phenotypic reservoirs. Another important element in virus evolution are bottlenecks. And bottlenecks mean that out of a very large number of genomes, you can, for example, transmit some of them, one of them or a few of them, as indicated here by the arrows. And I put these bottlenecks of different intensity. Okay? And that has several implications. It has a power to diversify the virus further, what in population genetics, you would say it increases the genetic drift of the population, and that's one way of diversification also, not only selection, but genetic drift. But also, we know now that individual genomes from the same mutant swarm may interact, often through their expression products, and give rise to positive interactions. For example, complementation, that has been done now with many systems, but one of the pioneers was the group of Shirogane and colleagues in Japan working with measles virus, or negative interactions of interference, of which I will talk later, again, in connection to lethal mutagenesis. So bottlenecks are not only a way to increase genetic drift, but they may also relieve some viruses from such interactions, and therefore, if a virus was suppressed because of this, for example, interference by the rest of the genomes of the population, the bottleneck may eliminate such suppression and manifest a new phenotype. So bottlenecks are important. And another thing that happens with bottlenecks is that they may dictate increases or decreases of fitness. What is fitness? Fitness is the capacity of a virus to replicate and produce progeny. Okay, so again, work that we did a long time ago with John Holland, and many people have done this. We know now that if you allow a virus to multiply, it is common sense. You keep selecting the viruses that replicate best, and that gives rise to a fitness increase, left of your screen. But if you introduce, for example, repeated bottleneck, fitness goes down. Why does fitness go down with bottlenecks? Because bottlenecks fix mutations, as indicated in the scheme on the right, in which you see these blue, red square and yellow square that have been fixed because of the bottleneck. And one of the general principles of genetics is that for any organism that is reasonably adapted to an environment, the probability of a mutation being deleterious is always higher than the probability of a mutation being beneficial. So the end result is that bottlenecks, due to the accumulation of mutation, result in fitness decrease. And fitness is very important because of several reasons, and you'll see that later, because fitness influences, for example, the response to antiviral treatments, but that comes later. Let me summarize what I have said until now. Summary of concepts: molecular and evolutionary basis of quasispecies dynamics of RNA viruses. The first and important point is high mutation rates. 10 to the minus 3 to 10 to the minus 5 substitutions per nucleotide due to absence, in some cases presence, but poor efficiency of error correction. And that means that the error rates are 1 million-fold higher than those that have been calculated for cellular DNA. Second point, there is rapid genome replication and high virus turnover in infected hosts. Viruses renew a lot in infected hosts. Then comes the essential conceptual point, Darwinian evolution. And Darwinian evolution can be divided in 3 points: genetic variation through mutation, recombination, and reassortment, competition, and selection. Okay? So these act on the virus populations in the same way as they act in all the organisms, except that for viral populations, the time factor has been compressed. So what takes millions of years in the evolution of a higher organisms, insects, simians, whatever, it takes minutes or days to witness evolution in the case of RNA viruses. Okay? I mentioned already there are interpopulation interactions, complementation, cooperations, and interference, and the bottlenecks event that I have defined. And I'd like to spend a few minutes because what we learn about viruses can have also some impact on other diseases, for example, cancer. And the reason for that is that some of the basic concepts apply also to cancer, eukaryotic parasites, bacteria, and even prions. I will not extend about quasispecies in prions, although this has been documented also quite clearly. In the case of prions, quasispecies are not due to mutations, but are due to conformations of the prion protein, minority conformations of the prion protein. But that's another story. I will not have the time to go into that today. So is there some parallel of what I am telling you in the parallel world? And the answer is yes. And let me introduce this because it has implications, as I said, for cancer, for example. Cells have error-correcting mechanisms to prevent or eliminate mutation and other lesions in DNA, proofreading repair activities, 3' to 5' exonuclease, and several post-replicative repair pathways. to give you an idea about the 100 proteins encoded in the human genome are involved in maintaining DNA integrity. That means that this is important. Okay. Now, despite this, lesions in DNA occur, and some are associated with disease. Single-cell analysis, transcriptomic, proteomics have documented cellular heterogeneity, that is, trait variations among individual cells within a tissue. Its functional significance is still largely unknown. And cellular heterogeneity due to genetic and epigenetic factors is accentuated in tumor cells, and it is a major determinant of tumor adaptability and resistance to treatments. And I have drawn this schematically showing that the same as in the case of viruses in which a bottleneck can drive one particular genome into a new quasispecies, the same with tumor cells. Tumors are heterogeneous, and one cell from a heterogeneous tumor can give rise to a new tumor with a different color that its cells become heterogeneous. And in these articles that I have put, mainly the article by Hayford et al., there are many other studies on this now. This is a recognition that the adaptability of tumors is really essentially due to heterogeneity. There are other factors, but one is heterogeneity. So there are genetic, epigenetic, and stochastic factors that influence tumor heterogeneity with consequence for cancer treatment failure. And again, on the same topic, intratumor heterogeneity has been related to tumor progression, metastasis, and treatment failure. Clonal heterogeneity in cancer is influenced by genetic and epigenetic factors. As you know, DNA methylation, histone phosphorylation. Drugs, anti-tumor drugs prescription based on the Genomic Data Commons Repository NCI is one of the recent initiatives. Cancer genotyping is carried out to quantify genetic heterogeneity. Of course, less is known about phenotypic heterogeneity, the same as with viruses. And many experimental studies are being approached with organized cultures, although these are expensive and difficult experiments. And the situation, of course, with cancer is much more complex. There are many types of different mutations that can drive the initial tumor, mutations that help maintaining the tumor, mutations that are adequate for metastasis or not. But apart from this different complexity, I want to emphasize the same principles of the dynamics that are worth understanding and studying, because they can transfer knowledge from virology to cellular biology and vice versa. Now, let's go back to implications for disease control in terms of what is the effect of quasispecies. I mean, if viruses were not quasispecies, they were just single sequences waiting there, to be controlled, the situation will be very different. But unfortunately, this is not the case. You all know that we have several approaches, and this is just a textbook slide, sorry, but prevention by vaccination, we have two very clear cases of success in the medical fields. For example, the eradication of smallpox or the animal virus rinderpest due only to vaccination. So these extinctions, eradications, were the results of human intervention through vaccination. So that's very important success. However, there are limitations to develop new vaccines, in particular insufficient understanding of correlates of protection, and of course also antigenic variation, and this is where quasispecies comes into the picture. A similar thing can be said of antiviral agents. Antiviral agents have not eradicated diseases, but there have been important successes with HIV-1 and hepatitis C virus, as you very well know. And of course, in veterinary medicine, we have alternative prevention of virus transmission and spray by culling of infected animals and their contacts. That has happened recently with SARS coronavirus 2 and some of these bison farms, and as you know. And of course, one has to also mention public information and political action, which is very important and behind also many of these problems. Now, let me give you some data that we have obtained with hepatitis C virus before we started working with SARS-CoV-2. And it will illustrate some of the points I am talking to you about. Well, you know very well, hepatitis C virus, about 3% of the human population is infected. 70% of the infections evolve towards chronicity, which is a risk for liver cancer and indication for liver transplantation. There is no vaccine. Now, the new therapies with direct-acting antiviral agents are extremely successful, more than 95% sustained responses. But of course, we have still problems, possibility of hepatocarcinoma recurrence, selection of inhibitor-resistant variants, hepatitis C diversification in the field that has not yet happened, at least that I know in a very clear way, but it's a danger that we have, and a lot of political problems behind this, as usual, okay? Many infected people remain undiagnosed, and treatments are expensive, not always available. So the possibility of global eradication depends on extending programs for diagnosis, treatment availability, and on resistant mutations not becoming epidemiologically relevant. An understanding of basic evolutionary mechanisms is possible with cell culture systems of hepatitis C infection, and consequence of extensive virus passes in an invariant cellular environment has been studied by us. What we did was we obtained from Charles Rice, a very important Nobel Prize and also very generous guy. He gave us the plasmid to express the infected virus. This is shown here schematically. And one thing that we address several questions. One of the first one was, can we detect specific interferon-resistant mutations? And that was not successful. We obtained populations that were resistant to interferon, but it was a kind of different types of combinations of mutations in different genomes, very complex. But there is another point that we addressed, and the people in my lab that, of course, I will acknowledge at the end, were extremely patient to give 200 passages in cell culture, as I mentioned at the beginning, 200 passages of hepatitis C to obtain a virus that in rat environment, in human hepatoma cells, had increased its fitness by about 2.2-fold. And then what we found with this high fitness virus is that the response to treatment was not the same as with the low or the basal fitness virus. And this is shown here for several drugs. Well, the controls with no drugs as usual, but we tested interferon, ribavirin, telaprevir, daclatasvir, cyclosporin A, sofosbuvir, and in all cases, you can see that high fitness gives resistance to the treatment. And that was something that we didn't know. It could be suspected, but we had to show it. And we showed that, and at the same time, we then said, then let's look at what alternative treatments do we have if a thing as simple as that a high fitness is already sufficient to render inefficient the treatment with an antiviral agent, what can we do to really control infections in a better way? And review of the literature and many facts that you know very well, I will not give details, but there are a number of strategies that one can follow. Combination therapy is obvious. Nowadays, people know that if you give monotherapy for a viral infection, you really risk very strongly that you are going to select resistant mutants. There may be examples of not, but the general rule is that viruses know the way to select for resistance. But if you look at the literature, people have tried also splitting into an induction and a maintenance regimen, although this has not yet reached the clinical setting, targeting of cellular functions that people thought it would be a way out of the problem of escape mutants. It may be a way, but it is also a way to get, side effects with the drugs and so forth. Then there are people are studying innate immunity response stimulating drugs, for example, inhibitors involved in pyrimidine biosynthesis. Don't ask me the mechanism. I really don't know. Nobody has clarified it to me. People have proposed and are now again trying to use combinations of immunotherapy and chemotherapy. And the strategy that we decided to work on is lethal mutagenesis, which means extinction of viruses by excess mutations. Why did we decide to work on this? Because essentially we like it, and because it is a consequence of understanding the quasispecies dynamics. In fact, Manfred Eigen and Peter Schuster already proposed the entry into error catastrophe as one of the consequences, or one, if you want, of the corollaries of quasi-species dynamics. And we like the concept, and I think that overall it is being reasonably successful. I have a bit of anecdote that I think I have time to tell you. When I wanted 20 years ago or even more, start working on lethal mutagenesis, I asked two important people what they thought. One was my previous mentor, Charles Weisman, and he said, "Oh, Esteban, it will certainly not work, but you will learn a lot in trying." And the other person that I consulted was Manfred Eigen, the great Manfred Eigen in, the one of the quasi-species theory. And he said, "Oh, yes, Esteban, that's fantastic. Certainly it will work." When I saw that two people so important had these slightly different views, I said, let's go ahead and do it. So we did it. And I think it has been reasonable. Of course, things take time, and there are obviously some problems with lethal mutagenesis. I cannot deny it. But let me tell you what is the concept. It's schematically explained here. And you have worked a lot on this. If this slide translates the quasi-species theory in a way that can be applied and seen easily in terms of virology. If we imagine fidelity 1, one extreme is fidelity 1, or totally unrealistic. Everything is perfectly copied. If that were the case, we would not exist. There would be no evolution on Earth, okay? And the other extreme is fidelity 0, which means that in genetic messages, anything can be copied into anything. Again, unrealistic, because if that was the case, there would be no information to maintain. But what happens in viruses that it fits the theoretical concept is that you have a quasi-species distribution, as I have been mentioned until now, when you have a certain fidelity. But when the fidelity goes down, and we can do that by antiviral agents that decrease the copying fidelity of the polymerase, okay, we start having a phase that we call lethal defection. That is interesting because the excess of mutation creates defective genomes that are interfering. Now we see one of the consequences of these mutant spectrum interference. Lethal defection consists in generating mutants that interfere with replication of the whole population. Then we have what I call overt lethality, and that allows us to cross an error threshold, which means that everything becomes random sequences of, if you want, there is a kind of an earthquake in these lines of genome distribution. So the error threshold is a maximum mutation rate at which the dominant master sequence can stabilize the mutant ensemble, and lethal mutagenesis is defined as virus extinction by an excess of mutations. in the references I gave you, you will see these concepts. There are others, of course. And perhaps just to mention that Larry Loeb, in that paper in Nature Review Cancer, already many years ago, already anticipated that lethal mutagenesis could also be used for cancer. And in fact, some of the cancer agents that are being used now, this is a growing field, have to do with trying to enhance the tumor heterogeneity to the point of making the tumor lose conceptual the possibility of being stable as a cellular ensemble also. So the concept again has some application nowadays being explored for cancer. How do these antiviral agents that increase the mutation rate of RNA viruses? They essentially work by mispairing, creating wrong base pairs so that the mutations are generated, and when the analog is incorporated as a template, it can also generate mutations. I have shown here the example of fluorouracil that is not used for viral therapy, yes, for cancer therapy, but clearly ribavirin or favipiravir are used as anti-cancer agents. They have, as antiviral agents, they have several mechanisms of activity. So I don't claim, obviously, that lethal mutagenesis is the only activity. This is not the case. In fact, ribavirin has many mechanisms of activity. One of them, in some cases, is lethal mutagenesis. And when it is lethal mutagenesis, is through base pairing, as you can see in the slide. So a typical old experiments, one that I don't know even if it was ever published, but I have it as a slide to illustrate the point. For example, hepatitis C virus. This is a standard hepatitis C, the low fitness virus. If you passage the virus without drug, the virus keeps replicating in human hepatocyte cells, a total number of 5 passages. If you add ribavirin, after looking at what are the concentrations that may work, ribavirin can extinguish the virus, and favipiravir can extinguish the virus. Extinguish the virus means that the virus goes below the limit of detection, and we have a protocol that it's in our papers. We have to do blind passages and lose RNA, lose infectivity. I mean, there is a protocol. It's not that just we lose the infectivity. We have a protocol to be sure that when we say that the virus is extinguished, at least it is below the detectability of infectivity and PCR the way that the technologies stand now. And a decrease of a specific infectivity, in other words, the ratio between infectivity and amount of viral RNA precedes extinction. Here it's written, a virus is considered extinct when no intercellular extracellular infectivity of RNA is detected, including after blind passages in absence of drug. That's important because, sometimes in the lab you can lose the virus for many reasons, but you have to be strict to be sure that you have evidence of extinction. And I'm going towards the last part of my talk. We found something interesting that has opened now a kind of research topic for us that we call synergistic lethal mutagenesis. And we define as synergistic lethal mutagenesis the one that results in synergism between 2 drugs, at least one of which acts by lethal mutagenesis. And the first interesting thing was favipiravir and ribavirin both target the polymerase for hepatitis C, but both exert a synergistic activity despite targeting the polymerase. And if you look at the textbooks of pharmacology, you will see that people say, 2 drugs will be synergistic when they target different points in the infectious cycle. But of course, there is one easy interpretation of that, and that is that favipiravir and ribavirin, despite targeting the polymerase, first, they target the polymerase in different ways. For example, ribavirin is not a chain terminator. Favipiravir has an activity. And another thing that we have found is that the place where they introduce mutations is not the same with the two agents. So that even that, if they were acting only through lethal mutagenesis, it is likely that we still would find a synergy because they have a different choice of the mutation point. Why? We don't know. But you see in the panels, you see an interesting result that was the one that was encouraging to us. We compared these activities with the low fitness hepatitis C, passage 0, high fitness passage 100, and high fitness passage 200. Passage 100 and 200, they already reached the maximum fitness, that 2.2-fold that I mentioned at the beginning. And you see the way to get the high fitness virus extinguished was through the combination of ribavirin and favipiravir, which is in yellow, the yellow line. If we use only favipiravir or ribavirin, the virus, the high fitness virus, is not extinguished. The low fitness, yes, but not the high fitness. And well, on the bottom left, the way we do that now is we put into the computer, the program is called Compusin, and you put the result of different mixtures of the two antiviral agents, what is the titer of your virus that you obtain in the progeny, and then you have a graph where you can distinguish antagonism from synergisms and from additivity. So that was the first example with hepatitis C of synergistic lethal mutagenesis. Now, with the emergence of COVID-19 in the human population, similar questions on antiviral interventions can be posed for SARS coronavirus 2. And these are questions that are now that interest us now a lot. Does this coronavirus display mutant swarms despite being a 29 kb genome and encoding an exo-N activity that should restrict mutational input? Do we have to fear less the impact of quasispecies dynamics of SARS-CoV-2 regarding vaccination or antiviral treatment failures? Is the future of SARS coronavirus 2 going to be closer to that of influenza, hepatitis, HIV, or FMDV? This is foot and mouth disease virus, okay? One virus, my favorite virus from my young times, as opposite to rabies or measles, as initially suggested. That answer I can already give you, the last point. It will be much more similar to influenza, certainly, than to rabies or measles and so forth and on, for sure. I am now totally convinced. I was not when I wrote that slide a few years ago, but now I am convinced. Now, you know, SARS-CoV-2 is evolving, many clades replacing one of the others. A rate of evolution, that's not the mutation rate. The rate of evolution that we calculated in the lab as average of 10 determinations, very similar to that of other RNA viruses, very similar, 10 to the minus 3 substitutions per nucleotide. And here is something interesting. Here we have a complex genome, right? And the theoreticians and people say, well, you know, variation will not be so important. So let me tell you the following. The complex NSP12, NSP8, NSP7 that we are studying now, also in collaboration with our colleagues in Barcelona, Núria Verdaguer and her team, this is the minimal polymerization complex, which is active in vitro. Now, NSP12 and NSP10 with NSP2 includes a 3', 5' exonuclease and a methyltransferase activity. That exo-N, if it behave as in other coronaviruses, such as the murine hepatitis virus, it would lower the error rate by about 15-fold. This is the work of Mark Dennison, as you know, and so forth. Now, some measurements attribute to the SARS-CoV polymerase complex a fidelity of 10 to the minus 1 to 10 to the minus 3 misincorporation per nucleotide, which is much lower fidelity than other RNA-dependent RNA polymerases. Some that we work on foot and mouth disease virus, the poliovirus work, a lot of work, and that was published last year in Journal of Molecular Biology, Ian et al. And it means that even if there were a correction of errors of about 15-fold, this virus has the possibility to mutate as much as any other RNA virus, despite being a coronavirus. So I summarize a methodological. We wanted to then, I know you have done work on this also, we wanted to penetrate into the mutant spectrum of isolates of COVID-19. So we have applied the Illumina MISEC platform and as via informatics pipeline called SIGDIP to identify and quantify minority mutations and deletions in the mutant spectrum of the NSP12 and the S coding regions. They are of interest to us for obvious reasons, which represents 7.8% of the genome, but we have reached a 0.1 mutation frequency percentage of frequency cutoff. That is to say that we can trust reasonably. Never in biology is everything absolute, but we can trust reasonably that 0.1% frequency cutoff. That is explained. This is due typically to the clean coverage that gives a quality score, and then we have done several controls, mutations that we determined with 0.5% and 0.1%, so similar mutational biases, distribution among the three codons, representation of the reduced amino acids in the GSID data bank. Duplicate sequencing runs gave a robust similarity in mutant spectrum and mutant number and frequency, and this has been clearly stated in our publications, essentially because reviewers force us to be very careful with this. But the panorama is amazing. If you look at the mutant spectra, these are the mutant spectra, just so you have a glance of what happens. Each of the little yellow squares is a position where you have a mutation. And there are also deletions, and there are some that the few ones that you see darker means higher frequency, okay? But the yellow ones are between 0.1% and 0.5%. So it says here with a 0.1 frequency cutoff, 38% of positions are mutated in these isolates, 38. I know there are defectives, there are viable, whatever, but 38%. And 97% of the mutations are represented in the consensus sequences of data banks, so that they are unlikely to be artifacts for this and other reasons. So we then analyzed, and that's some very recent work, the paper has been published just a couple of months ago. We have derived two-dimensional self-organized maps to visualize nucleotide distances among haplotypes with haplotype frequencies as the third dimension. You'll see now the plots. These are called self-organized maps. If you want, it's an application of, I don't like the term, it looks a little bit far-fetched, but if you want artificial intelligence to the analysis of mutant spectra. And the objective with this procedure is to compare intra-host and inter-host sums and to test whether the incipient diversification could have functional relevance. And I am going towards the end of my talk. This is the type of plot that we have. The paper, it's here if you want to see the details. So each map includes what we call a master neuron, which is the high peak. That mountain that you see, sharp mountain, is a neuron. The neuron contains some different haplotypes, not necessarily only one, but haplotypes that are very close to one another. And you can see the cloud, the mutant spectrum cloud, that was those lines that I showed you at the beginning of my talk, now becomes that field of little blue little blue peaks, right? That we can compare location, distance, and so forth. In fact, in here, in that paper, we distinguish three subclasses according to mutations in the master neuron relative to difference with the isolate Wuhan HU1. And not surprisingly, to those of us who are believers in quasi-species, each sum is unique to one isolate. And with our colleague Nuria Verdaguer, we showed, I will go rapidly because I don't want to take more time. We showed that some of these mutations that appear in the small blue peaks that are incipient diversification in a patient are functionally relevant because they can modify the activity of the NSP12, NSP8, NSP7 polymerase complex in a primer extension assay of RNA viruses in vitro. And similar prediction of functional effects are made with incipient substitution in S, although this is based on in the literature. We did not do work on that. So what can we do now to confront COVID-19? So now we go back and we say the dynamics appears to be very similar to the previous RNA viruses. So I put here SARS population dynamics appears similar to that of other RNA viruses. However, this point is we are still investigating similarities, differences with RNA viruses. So the challenge to develop effective antiviral interventions for COVID does not seem substantially different from the challenge we confront for other RNA viral diseases. And people are investigating a lot now combination therapies that are synergistic. And we did the same, and we found, and that has been, again, very recently published, that remdesivir and ribavirin can extinguish SARS coronavirus 2 at certain concentrations. They are indicated here, and we have the same criteria of extension. And this extension is essentially associated with synergistic interactions between the two. And that's shown here, remdesivir and ribavirin, same idea that we did with hepatitis C and ribavirin and favipiravir. Here again, we have synergy. We have not yet done any work with animals. They are too expensive for us. We don't have the money and we don't have the, let's say, the people to do it, although we could. We are trying to look for funding. And in this case, synergy is supported by two different computer programs, the Composine and another one that is called Zip Synergy, which with the same data, the input is the same data, but both give rise to the conclusion that we have synergy. And it's one of the points that encourages us to really pursue lethal mutagenesis for COVID-19 with new antiviral agents and new nucleotide analogs. So I come to the end of my talk. Viral quasi-species are complex and dynamic mixtures of variants subjected to competition, selection, and bottleneck events. A mutant spectrum may hide subsets of genomes with a different potential behavior, including, say, mutants to antiviral interventions. Mutant spectra promote adaptability to new environments. They exhibit parallel features to tumor cell heterogeneity. Self-organized neural network marks provide a means to establish relationships among haplotypes in mutant spectra and to identify incipient functional diversification of viruses. And synergistic lethal mutagenesis, remdesivir and ribavirin for SARS-CoV-2 offer prospects for broad-spectrum antiviral intervention. And of course, I have to acknowledge many people. In the picture, the lady sitting near me is Celia Perales, fundamental for this work. The lady on the right is Maria Eugenia Soria, fantastic person. Isabel de Avila in the middle spends essentially half of her life in the P3 laboratory. And then we have collaborations that in bioinformatics, Soledad Delgado, for example, has done the self-organized maps, and a lot of things that are going on. And we owe also a lot to the help of Luis and Juanes, coronavirologists, famous Spanish coronavirologists, who has taught us, shown us all the procedures so that we were able to start all this. So again, I thank you, all of you, for the invitation. I'll be happy. I don't know how you do this. If there is time and you have some questions, I'll be happy to respond. Thank you very much.
Thank you.